Yes, Dr. Kamna Misra. I think we can start now. Dr. Matlub Hussain.
Hello.
Hello, everyone. Yes, Dr. Kamna Misra. I think we can. So I'm going to start it now. Respected Principal Dr. Balraj Thapar, esteemed faculty members, and my dear students, a very good morning to all of you. I, Dr. Indu Mehta, Head of the Zoology Department, welcome you all in today's international webinar, which is being conducted by the Department of Zoology, GGDSD College, Chandigarh, India. May I request respected Principal Dr. Balraj Thapar to introduce today's speakers. Over to you, sir. वॉइस नहीं आ रही.
The number you have dialed is currently busy.
कैसे बोलूं? Voice.
Good morning, ladies. It's Namaskar.
Not visible, not audible. Okay, he can come later. सर का बाद में कर देते हैं फिर, है ना?
Topic as per the call of the time and in the backdrop of COVID-19 vaccine.
No, it's good.
Persons with us, namely Dr. Mrs. Kamna Misra from New Jersey, USA. She's Kamna Misra and Dr. Matlub Hussain, teacher and are amidst the resource person of today's webinar. On behalf of the management, the staff and the students of Goswami Ganesh Datta ST College, Chandigarh, India, all a hearty welcome. Today's international webinar is being conducted by the Department of Geology, GGDSD College, Chandigarh. The theme of the webinar is Therapeutics and more, which is very relevant topic as per the call of the time and in the backdrop of COVID-19 vaccine. Today we have two, namely Dr. Mrs. Kamna Misra from New Jersey, USA. She is the founder and chief executive officer of Therapeutics, New Jersey. She is the president of Association for Women in Science, New Jersey, USA. Our second resource person of today's webinar is Dr. Matloob Hussain of Auckland, where he's working as an associate professor in the Department of Virology. I'm grateful to them for not only sparing their valuable time, but their time. Their words of wisdom will be a source of inspiration and stimulation for all of us. Thank you. Thanks a lot. Over to Dr. Indu Mehta.
Thank you very much, sir. Now I will introduce today's first speaker. It is my privilege and pleasure to introduce to you the first speaker, Dr. Mrs. Kamna Misra. A neuroscientist by training, Dr. Misra has a deep interest in translational science to address the unmet social needs. She leads the R&D activities of a startup, ContraRx, a company developing tools to educate users about unintended harmful consequences of polypharma. She also had several scientific research roles, including leading the research and development activities of regenerative medicine, developing stem cell therapies for the spinal cord injuries. Dr. Misra has several peer-reviewed publications as well as socio-scientific and educational commentaries in the leading journals. Furthermore, she leads the Association for Women in Science New Jersey activities as a chapter president. She has spearheaded several initiatives for the New Jersey Women in STEM. Her initiative, providing entrepreneurial alternatives for New Jersey women in life sciences PhDs, designed to encourage women to embrace entrepreneurial ventures, was funded by National Science Foundation. In the recognition of contributions, she is a member of several boards and is a frequent invited speaker at multinational and national venues, including the International Day of Women and Girl in the Science Assembly at United Nations headquarters. Well, for this webinar, she will be speaking on the topic COVID-19 highlights sex inequality in the drug development and therapeutics. Well, ma'am, participants are eager to listen to you. Now I request Dr. Kamna Misra to kindly deliver her talk. Over to you, ma'am.
Thank you so much, Dr. Indu Mehta, for the very kind introduction. And thank you to the esteemed principal also. I think this is a great initiative taken by the college as well as the faculty to sort of increase the interaction between the students and, researchers placed in different fields of science all over the world, because I think this is something that only the unique situation we are in today allows us to do so easily with the facility of webinars. So I'm very thankful to all of you. As per my belief, based on what Dr. Mehta had told me, I think we are speaking to a set of students who are mostly in the undergrad stage, I think in the zoology background. So I have tried to keep my talk very structured to a very basic format, just talking more of the journey as a scientist, and I will leave the technical details to my colleague who is coming soon after, Dr. Matloob, and I think he will give a very detailed instruction on the science part of it. What I'm going to do today is tell you how my journey began right where you guys are today in Chandigarh in Punjab University. I was studying at one point with Dr. Mehta when we were in zoology department honors school a while ago. So, I will do a brief introduction of what I have done, where I am, what I have enjoyed. And I do hope and encourage all the students to participate and ask questions if and when they need to. And feel free to reach out later on. I'm sure Dr. Mehta will be able to share the contact details, and happy to sort of go from there. Dr. Mehta, do you think I'm in a situation to share my screen? Yeah, I'll try.
Yes, sure.
Okay. Let me get my glasses. Can you see?
We are just waiting. Yeah, it's visible now.
Okay.
Kamna, it's visible.
Yes. So, I will just give a brief introduction of who I am, what am I doing, where I am. So, I had training in viral oncology, both for my PhD, which I had done in Punjab University itself. At that point, the Department of Biotechnology was just being set up. And I think I was one of the first few students sort of enrolled in the PhD program there. And during that time, I was working on hepatitis B virus, HPV, and its role in carcinogenesis and how it is involved in causing cancer, not cervical cancer, but the oral cancer. It was a collaboration. The work was done at PU, but there was a lot of work also being conducted at PGI. So once I was done with that, that sort of opened the window for me to get into international genetic. I think I should go to the next slide. Oh, sorry. Yeah. So that sort of opened the window for me to step into ICGB in New Delhi, India, which is the International Center for Genetic Engineering and Biotechnology. I think at that point, I was fortunate to really work with few of the best scientists India had to offer in the field. And it gave me a lot of opportunity to learn at that point, the current techniques and sort of cutting edge technologies in the field of virology at that point. And using that, I was able to leverage that to take me on to, I wanted to then carry on and do my postdoc. I thought US would be a good place to come to, and I came here. And at that point, I had an opportunity to transition from the virology field into the neuroscience field. It so happened that the lab that I chose was doing that kind of work. I ended up studying neuroscience, I think, for majority of my life now. So I always call myself a neuroscientist, not a virologist. During this period, I was working with medical school, which is situated in New Jersey. In addition to research, I had involvement with teaching anatomy classes, etc., in the medical school, and sort of continued the same field. And in neuroscience, also there are subdivisions. So from spinal cord to retina development to brain development to neural disease, sort of had an opportunity to play with a lot of these things during that time. So during my period at the International Center for Genetic Engineering and Biotechnology, as I mentioned earlier, I got an opportunity to play hands-on with a lot of that time cutting-edge techniques. I think we were the first few labs which had transgenic animals, in those times, and really some good sort of tools to analyze data from that. Pouring our own sequencing gels, PCR was not so bad, and then the other kind of assays. And having worked on these things, I had a skill set which I could offer to the labs when I started applying for positions in the US. I had a kind of something in my bag that I could offer to a lab that, hey, I know this, and it would be a good fit to sort of take it from there. And based on this, I ended up as I mentioned, into the neuroscience field. What I focused on for the next 10 to 15 years was therefore more of developmental neurobiology. It's studying the cell biology of neurons, how neurons are born, how neurons differentiate, how do they perform different functions. And then obviously, like any other field, once you know the physiology and anatomy of a structure, you also sort of experiment with what happens if things go wrong, right? If you have neural diseases or if you have spinal cord injuries, what are the various aspects that you can address there? Broadly, we all know the nervous system, two parts, the brain and the spinal cord, right? These two are enough to do a lot of activities. I think I'm going to try to play this movie. And Indu, I invite you to ping me if you feel this is not a clear transmission. Just watch this movie
right.
first.
Sure.
Just watch it, and then I will take it from there.
Okay. It's perfectly fine.
You don't know this young woman, but whether you want to or not, in this very moment, you've already passed judgment on her. In a split second, you've already decided if you find her trustworthy, competent, likable. No doubt your brain is a state-of-the-art marvel, managing 90% of everything you do without letting you know, regardless of whether you're awake or asleep. Something in your head navigates you through the everyday adventures of modern life. Something that decides things for you before you can think about it. Because your brain is always on automatic.
We're driven by our unconscious mind. Isn't that the interesting thing? So much of what we do is unconscious. The decisions we make are almost dictated to us. I often wonder who's in charge here. Who's running the show? How much control do we really have?
So just sort of to show you the magnanimity of what our nervous system is really capable of. And, a lot of activities that we do in our life are conscious, but majority of the activities we are performing are unconscious. We don't even think, when we are breathing or when we meet someone, our hand sort of automatically goes and we shake their hands. We are making some effort, but a lot of it is sort of unconscious. So all these actions, the conscious, the unconscious, is sort of controlled by this nervous system, the brain and the spinal cord. And there are two main sort of tracks. I don't want to go into the neuroscience of it, but basically what we want to understand is that in this nervous system, this whole functioning is happening by different kinds of neurons, right? If you see on this, I don't think I have a cursor to show you, but on the right-hand side, you can see these colored neurons as you can see with different colors. There are thousands and thousands of neurons that we have in our brain and in our spinal cord. And obviously, these are different types. They have different projections. They have different functions. And all of these are developmentally controlled. And this is what developmental neurobiologists sort of studies, that how are different neurons born? How do they function? And so I'll show you in this slide, for instance. Let's go back to embryogenesis. Neurogenesis is kind of very similar to embryogenesis and other organ sort of regeneration, right? So the whole thing is when an embryo is born, it's a one cell egg and a sperm sort of fusing to make a single cell. And then these single cells will divide to make 2, then 4, then 8, and sort of the number grows exponentially. At this point, everything is undifferentiated. Each cell has a capability to form the whole body. You cannot say that cell A will make the heart or cell C will make the lungs. It's not decided at that point. But what happens is a little later at the stage, you see that 3 different layers of cell types are formed: the ectoderm, endoderm, and the mesoderm. And this is where the differentiation of different organ types really begins. It's very early on in the development, but this is where the foundation is laid that, this body will have organogenesis, and then different types of organs will be born out of different regions. So the nervous system is usually, it is born out of the ectoderm, right? And if you see in this figure, the blue and the green, you see a little turn of the ectoderm at a certain cell stage. It's like 3 layers, if you can think of 3 pieces of cloth lying on top of each other. There's an ectoderm, then there's an endoderm, and then there's a mesoderm. So the ectoderm sort of invaginates, sort of hollows out a little, and that's where the whole story begins. So one thing to remember from this is that early on in embryogenesis, there's very little differentiation. But as the cells keep dividing, they keep differentiating, right? And the stemness of them is decreasing, and these cells get more and more. And once the cell is sort of differentiated, it cannot usually come back, and it just goes to a differentiate. And this is where a very big phase of our medical research, the regenerative medicine, is sort of based on, and that's why I'm sort of mentioning this to you guys. So let's talk about neurogenesis. Each one of us has like almost 100 billion neurons, and majority of these neurons are formed before you're born, and some of them do develop a little after you're born, maybe a year or so. But by the time you're a few years old, those are the maximum neurons you can have. And by the time you're in your 20s, they in fact start dying. And this is the whole issue of sort of the nervous system, that you cannot make new neurons, right? So if you have any injury, whether it's in your brain or it's in your spinal cord, it is a permanent loss forever. And that is why there's such a huge emphasis on, at least in these fields, to try to get stem cell therapies to work, because there is no other way you can address the shortcomings that happen. And just something out of interest, that when a child is born, the neurons by themselves are individual, right? You can think of them as balls sort of in the space. They're not connected to each other. And for the nervous system to be functional, these neurons have to connect with each other. It's not a physical connection, it's something called a synapse, right? So each neuron or a ball would be you're sort of communicating to the neighboring neurons, let's say 5, 6 of them, and that's called a synapse. So when a person is born as a child, initially it would have around 2,500 synapses per neuron. That's the amount of communication it's already doing. But as you develop, as you learn, as you sort of understand things, these neurons make connections, like more and more robust connections. And by the time you're 2 to 3 years old, there are 15,000 connections per neuron. It's like really communicating to all the neighboring neurons and trying to relay information. And these circuits or these synapses are what helps us build our memory and other functions of the brain. And it is also known that if you're not using certain circuits, these circuits are going to get deleted. And that's why, it's these memory games and all are helpful, because you're trying to strengthen your neuron connections. So how does this all happen? So on the right-hand side, you see this is an early embryo. This is one of my own research from earlier on, where you sort of stain it with, neurofilament, and you can see the whole spinal cord and the brain sort of darker than the rest of it. Although it looks very simple on the outside, but when you go inside, and this is what I'm showing on the left-hand top, I'm not going to explain. This is, not really the scope of the talk today. But within this early spinal cord also, when you open it up, there are already 15, 20, 30 different kinds of neuronal subtypes in there. Each one has a specific position in the spinal cord, and each one has a specific function. But if you look at the top figure, you see a central canal, which is a central part, which is empty. And then on the right-hand side, you see some colors. So what is happening is each neuron has to move within that space and sort of don a character that it's going to help the mature nervous system to sort of perform the functions, right? These are some of the figures that I have generated over last 15, 20 years and some of the publications we have done. And this is a fascinating field for someone to get in. You can get into the brain, you can get into the spinal cord. The left-hand side you see are these spinal cord sections, and the techniques used here are called immunohistochemistry, where you're staining with antibodies. These are live neurons, actual neurons, and you're using confocal or electron microscope based on what techniques you're doing to simultaneously detect in this figure up here, 3 different types of proteins, right? And in the center you see is a section of a retina. So this is an sort of retina or eye developing in the embryo, and you get an opportunity to look at what are the different factors that are secreted and then how the eye system matures. On the right hand side is a mature spinal cord which is injured, and you see the green stuff oozing out, and so there are many thousands and thousands of things you can do in cell biology. This happens to be related to neuroscience. All it talks about in this field is that, okay, there is a cell which doesn't know what it'll become when it is adult at a certain point. But then slowly it starts deciding that I have to go on to the next stage. So it's getting more and more differentiated. And as it is getting differentiated, it loses the capability to become what it was earlier. Earlier, it was a stem cell. That is a naive, actual stem cell. But then it's becoming a neuron, or then it's becoming a myocyte, or whatever special function. And this is happening due to certain factors that are being secreted in our body. So one factor comes on, it makes that naive cell into a slightly more mature cell. That cell then secretes more factors, another signaling pathway or a cascade is activated, other genes come into play, and it sort of gets differentiated. I have tons and tons of publications in this field. Those are all very exciting and very interesting, but what happened with this was, as I've been trying to make you aware of stem cells, I keep talking about it all the time, that cell biology and developmental biology is sort of very well connected to the stem cells. I got a little interested in the stem cell aspect of it because, as I mentioned earlier, neurogenesis is something, once you have a spinal cord injury or you have some kind of a, brain injury or a brain disease, currently, there are no cures at all. And it has been, unfortunately, for the last 50 to 100 years, so many drugs, so many pharma companies, so many biotech companies have been trying to get therapies. It's not like they have not tried. But for whatever reason, maybe our approach is so wrong that nothing has really worked. You have cures which can delay the, sort of the symptoms, but there are no cures as such. And it is my own personal feeling based on my experience with whatever stem cells I have handled, and a lot of other people in the field, that actual sort of cure would be existing in these stem cells if we understand how to manipulate them and use them. So if you look at the nature, as the embryo is developing, what is nature doing? It's taking a stem cell, a naive cell, and it's making it into, let's say, a neuron by 10 steps. If we know these 10 steps, that A becomes B, B becomes C, C becomes D, and then a neuron is born. Now, if this neuron is injured or damaged and we want to repair it, we should be able to take a stem cell and do these 10 steps, A, B, C, D, and make this right again. So there is a lot of promise in this field. And in my personal opinion, in the next few sort of 10, 20 years, this is a field which is really going to explode because there's a lot of potential. Currently, we do not have the resources to really understand the, really in-depth biology of how it is happening. We are all trying. But this is where the field is sort of headed. And based on this experience, I was able to get associated with a startup company which was developing stem cell therapies for treating spinal cord injuries as a director for research and development. So what that showed me, science I had been doing for so long, right? It was already 10, 15 years since my postdocs and other kind of research. But when I got into the company aspect of trying to develop a therapy, right? So science is a big part, but that's not the only part. When you get into a company, you have to make sure that you're seeing the whole robust R&D portfolio growing. You have to execute agreements. You have to do licensing agreements. You go to an academic organization. You need their technology. You sort of execute. Oh, you get this. I get this. For this, we pay so much. You have royalties. So I was able to sort of get associated with the company as it was being born from an idea, from a patent at a university, and nothing else. And it needed a lot of effort. It needed, you had to wear every hat possible. You would be doing the research, you would be raising the money, you would be going and meeting people, you would be the face of it. But it also provided me such a unique opportunity to really learn that science has many other aspects rather than just doing experiments in a lab and teaching. You have so many other opportunities to really make a difference to people's life, right? And I think at that point I was sort of hooked onto it. Yeah, we did publish one or two, I published some papers during that. I got funded with grants to support that work. We had, as a company, conducted a clinical trial on 70 people with spinal cord injuries, right? And we had excellent results showing that this sort of the product that we were developing, which was autologous adherent bone marrow stem cells, is a good sort of way to go. But what also happened is that I got interested in this other aspect of science, which is the technology commercialization or early stage investment, right? So the reality is there are thousands and thousands of ideas which keep coming up in science every day, because science is, at least currently in academia, is you do research, you publish it, you get more money, you do more research, you publish it, you get more. It's a sort of a vicious cycle. You keep going on and on. But if you really want to take that technology out and invent it and make a cure or make a product out of it, you need a lot of money. It cannot be done without money. And so that's the aspect of commercialization of technology, right? So I have always been interested in that part of science. I tried initially as a postdoc to try to patent some of the things we were doing. I didn't really know how. My advisor was not super supportive of it, so it never went anywhere. But then when I was with this company, it sort of reawakened those it gave me the idea to see how it is actually happening. And it really opened my eyes, because you take a technology from an institution, there's a lot of money talk about it, then you have ownership stakes, who gets what, because of working for the company, you have so many sort of percentage, it sort of teaches you a completely different aspect of science, which as scientists, none of us have really exposure to. And then another aspect of it is how you get the sort of investment from venture cap groups, how do they take a share in you? Sometimes you as scientists just get kicked out of the company that you have raised as a baby, and stuff like that. And so I got associated with this venture cap, which had another 10 sort of portfolio companies where they were investing in, right? Someone has an idea, they take it to a next level, and then they take money from this venture cap group. So now the venture cap group has to make sure that the company is getting the tools to grow. Okay, they need money to do this sort of technology, to do cancer diagnosis. They have to develop a kit. The kit can be manufactured here for that, all those kind of things. And at a certain point, I thought, if I can do it for other companies, why can I not do it for myself? And that's how my own sort of startup concept was started. And this company that I talk about, it is in the space of pharmacovigilance, which might be a new concept for you. I will briefly mention what it does, but basically the technology that it is based on, I have licensed from Columbia University and Stanford University. So these two universities, the way it works now would be a part, they are partners in the company that we are trying to get, hopefully somewhere. And the problem is based on something called the drug-drug interaction or adverse drug reactions. So as more and more drugs are coming into the market, all of you might know, your parents or your grandparents or someone in your family who, let's say, is a high blood pressure patient. So they will be taking medications for keeping the blood pressure low. Okay, good, safe. The drugs have been researched, well, they're safe. But what happens is now they might also be having some other disease, let's say, arthritis or some kind of a seizure. So now they have another medicine prescribed to them, which is for seizure control. That drug by itself is very safe, but no one has ever done the research of what happens when the seizure medication is taken along with the blood pressure medicine in a certain population. So when we talk about drug-drug interactions, this is what polypharmacy is introducing, where people are taking more and more medications, and then it ends up exposing them to potential dangers of multiple drugs. So it involves the study of drug-related injuries and making warnings or withdrawal recommendations from pharmaceutical agents. So, you have to detect a drug-drug interaction, then you have to decide, is it a true interaction, and then sort of how do you prevent. So this is the concept of the pharmacovigilance. So what is happening? I'm just giving you some numbers in the US because that's where I am. So I try to research that market more than other thing. In US alone, around 170 million Americans use prescription drugs, right? Out of them, 119 million are at least on one prescription drug. 85 million are on at least two prescription drugs. 34 million are on 5 or more medications. So just try to imagine how much is the danger in this last group of people who are taking 5 or more medicines. Each medicine has been sort of developed in a isolated ball where it did not have a background of the patient taking other medications. This is a very sort of a new field in the sense that it's a growing field. Not too much focus has been paid till now to this, but it has serious consequences. This affects the economy in huge numbers, and I think there's a market for this, and that's why. But in context to, let's say, the COVID-19, because today's title somehow mentions COVID-19. Through our company recently, we have published this article in Frontiers in Cardiovascular Medicine. So again, what happens is that we all know that initially when COVID-19 sort of exploded, and one of the first treatments that people thought works based on a publication that came out in France on a clinical trial was that HCQ, hydrochloroquine, is a very good treatment could be helpful to the patients with COVID-19. And before it could be clinically sort of validated whether this is really true, all over the world, these medications, chloroquine and HCQ, were being given to patients because there was no other alternative, right? And slowly people started realizing that there are some health risks associated, specifically the cardiovascular risk, which is called the QTc prolongation. So it's like a beat of the, if you know the pattern of heartbeats, it's like it sort of elevates to such a level that it can ultimately lead to death because of elevated QTc prolongation. And we were very fortunate at that point, because since my company is trying to focus specifically on drug-drug interaction, and now you have the situation of COVID-19 where a lot of people are being given, chloroquine or HCQ, and they might have other sort of prescriptions going on along with that. So we were able to collaborate with the best clinical trials happening at that point. I think within 3-4 months, people who you see listed on this paper, Lior Jankelson, he's an MD PhD. He led the clinical trial at New York State, and one of the biggest study on this kind that was published from here. Then the next person, the Van den Borg, he was conducting another study in Netherlands, and they had come out with a study of how HCQ in their studies had really elevated QTc in the patients there. Martin Coeur, he was leading a study in France, and they had also published a study about the numbers of how the QTc was elevated with these drugs, right? So we had these, at first nothing, then suddenly everyone was being given this medication, and after a little fallback, and then you have these flurry of studies which are showing, hey, HCQ and CQ is actually harming patients, at least there's a risk. And as a pharmacovigilance person, we have already been trying to develop tools where we sort of thought that these drugs are more harmful maybe to females. Females, especially in the reproductive age, a lot of them take contraceptives. Contraceptives are hormones, but at the end of the day, they are sort of structures which your liver has to metabolize. So there's already a background of potential drug-drug interaction happening there. So this study was published, and it was an excellent collaborative sort of study. Within 1 or 2 months, we were able to get data from all these authors who were so sort of willing to openly share data, and some good things that have come out of this pandemic has been this extraordinary scientific collaboration that we have been seeing. People are sharing data like they never shared before. So, I ended up going from with virology, neuroscience, and now I'm sort of trying to promote pharmacovigilance. On the side, as something which is I'm very passionate about, is to promote, since my own journey has been such, right? I'm not really sure about the Indian market, but at least in the US and in a lot of Europe, the past few years, science has seen a paucity of funding, right? The money available for research is going down and down. More and more people are being trained than the need of the people, right? So there's this pool of hugely highly talented people who have been trained to do something, but then that thing is not there. So I feel there is a requirement for changing the way we educate and our concept of education, even at least for STEM professionals, right? Traditionally, STEM professionals have been sort of bred to either have their own labs or teach, and that's why most of the sort of training focuses on that. But I think the current situation, the lack of funding for really starting your own labs, but you do have the capability, you have learned the science, and you know what to do with it. I feel that entrepreneurship is a huge potential for people who have that kind of aptitude. So you have a skill, you have the knowledge, and if you like some kind of risk taking, this is a field that a lot of people can divert to. And I just feel that they just don't have the resources. So we are doing a major push to provide these opportunities, at least because I'm associated with an organization called Association for Women in Science. So there's a major push to provide these facilities to our members who have an interest, who want to do something. And we say, okay, here, let's do this together. Let's provide these resources together. We have got funding, for instance, the latest sort of grant that I got through NSF with one of the local universities. It was money given to us to help provide entrepreneurial alternatives for New Jersey women with life science PhDs, right? These kind of people who were trained but now had nowhere to go to. So instead of just doing nothing, why don't you use that and try to get a product into the market, and let's try to help them and give them the resources they would need to that. So that is one of the major things I do, in the advocacy side, and it is a very rewarding and kind of an eye-opener kind of an opportunity. If you guys have time, so being involved in those kind of activities gives an opportunity to again see science from a different perspective. It gives you an opportunity to interact with policymakers, to sort of try to influence them to divert more funding to this whole STEM sort of enterprise. So if you have an opportunity, we can do it later. I had a video here to showing what all we are doing within New Jersey to promote these kind of activities, whether it's with legislators, whether it's with policymakers, whether it's with academic organizations or small local and pharma biotech companies. And I think, okay, I will not show that, but as I always say, anyone who has never made a mistake has never tried anything new. And I think that is worse than not doing anything. Thank you very much. And I would love to hear any questions that you might have.
Yes, anybody wants to ask anything? Questions from the participants, please. The participants can unmute their mic and then they can ask the questions to the resource person. It is the golden opportunity you are getting, I think.
Hello, ma'am. This is Jyoti. I would like to ask you one question. Like, we gave hydroxychloroquine and chloroquine, especially in case of the COVID-19 patients, right? And we say there is so much gender inequality existing. So how we are differentiating the drugs we are giving to a male or a female, especially if they're suffering from this disease?
No, there is no differentiation as of now. So that's what we are trying to show, that we need to differentiate. Unfortunately, the way the things are, the way it happens is that it's based on body weight, and they just, it's like taken as a woman is a mini man, a smaller man, but that's not true. We have a different biology. So this is one of the, it was not being done. That's what our paper study was showing, that females might be at a higher risk, but there was no differentiation.
But when we say, when I used to study the papers, we say that women are more these women, they are suffering more from this COVID-19. So what is actually the biology?
No. COVID, actually, the frequency of COVID-19 was more in males, but that doesn't mean that the females who are getting it. So QTC risk is different from, this is a heart condition which happens after you have COVID-19. So overall, more men were falling sick with COVID-19, but once they have it, even the few females who have, they are at a higher risk of the QTC, heart conditions.
Okay, thank you.
Yeah.
Yes, anybody else? Anybody else?
Good morning, ma'am.
Nazdeep Kaur. This is Nazdeep Kaur from BSc first year. Yes, Nazdeep, you can ask your question, beta.
Ma'am, I wanted to ask if there are any side effects of stem cell therapy.
Side effects in what sense? So the issue with stem cells is that, if you're doing it the right way, then there will be no sort of side effects. But no product has been developed to date, which is sort of FDA, at least for spinal cord injuries and stuff, which is FDA approved, and therefore at a level where, there are a lot of, quacks getting up in every nook and corner and claiming they are having stem cells. Those are not stem cells. So no one can say what those stem cells will do. The reality is we haven't reached that stage where we can be very confident about our product, So what you see in each nook of India, that is not really a stem cell. That cannot be guaranteed. But if a technology is developed and if it comes into the market, it has to be. So for instance, what we were studying was bone marrow stem cells. So stem cells are various kinds, you all must be aware of it. You can make it from embryos, embryonic stem cells. You can make it from fat, adipose stem cells. That's a new field which is really growing. Everyone can use their own fat to retrieve stem cells from them. But what we were doing was adherent bone marrow stem cells. So you take the bone marrow out and then you take out the stem cells. So these adherent bone marrow stem cells are derived from mature, bone marrow. So it's not an embryo. Embryo is a stem cell. So it can grow into cancerous things. It has a higher likelihood of sort of growing. So stem cells are various different types. It depends which stem cells you're talking about.
So, okay, any other participant want to ask any question? Most welcome. I think no more questions. Okay.
Good morning, ma'am.
Yes, Tanvi again, another student of mine. She's curious to know something.
Ma'am, I wanted to ask that, as you have said, you have done biotechnology.
Yes.
So what is the scope of microbiology in research field?
Oh, yeah. So I will take 1 minute to tell, of my own experience. Everything I did, no one in my family had ever studied, beyond master's or PhD. No one. So it was just, I had that booth in my head, I want to do it. So I had no guidance, right? I did whatever came on my way and sort of did it. If I look back, now there are certain fields which need to change. Microbiology will always have some sort of applications. It is getting more and more competitive. What I feel is we need to start learning a lot of computational biology, you understand, bioinformatics with a lot of programming and different things. So these will always have application. But if you want to really differentiate and you want to get into cutting edge research, we have to stop doing what we were doing 30 years ago, and there are newer fields coming up, a lot of and I think we as Indians tend to be good in these kind of programs. While you're doing microbiology, also do other courses on the side, which train you for these other opportunities. That's what I would say.
Ma'am, what are those other courses?
Yes. So like, for instance, because of COVID, last 6 months, everyone's been working from home, including me. So I decided to do a course from my local community college on the side. It's called Maya 3D modeling. Has anyone of you heard it? Look at it. So it's something you can just sit at home and train yourself, right? It's just showing. So for instance, if I want to tell you that, okay, this A stem cell is going to become a neuron, it has 10 steps. So if I try to tell you in words, it's very difficult to explain. But if I can make a 3-dimensional picture, instead of drawing multiple, images A, B, C, D. If I can make a sort of an image where this cell is moving, turning, twisting, and then becoming a neuron, right? It's called 3D graph, sort of three-dimensional picture telling. It's so easy to understand science, and that's where science is going. Science is going into 2D modeling, 3D modeling, where medical schools are teaching online, where you can go into a computer like a game, right? The way you play video games. So do whatever you're doing, but on the side, keep doing some small courses which teach you other things also.
Thank you so much, ma'am.
So it had been a very interactive session. Thank you very much, Dr. Misra, for delivering such an informative, useful, and rewarding talk. It was a wonderful talk indeed. I'm sure that all the students must have benefited by your encouraging words. So, thank you very much.
Thank you. Yeah, I'll go off the video now so that you can pass on to the next.
Right. Thanks. Now it's time to move further and listen to our second eminent speaker. It is my great pleasure to introduce to you our second speaker of today's webinar, Dr. Matloob Hussain from the University of Otago, New Zealand. Dr. Matloob Hussain is an Associate Professor of Virology, Senior Virologist in the Department of Microbiology and Immunology at the University of Otago, New Zealand. After doing his PhD, he moved to USA for postdoctoral fellowship from the National Institute of Health, USA. And subsequently, he became a scientist in the Respiratory Research Institute, USA, way back in 2000. Then he joined as a senior lecturer in the University of Otago, New Zealand, in the area of his own interest, that is virology. Dr. Matloob Hussain has been studying the molecular interactions of viruses with human host to expand the knowledge base and design host-directed antiviral strategies. He has published 28 research papers in the journals of international repute. He has supervised over 20 research students and staff. He also teaches virology to the undergraduate and postgraduate students. He has very rich experience of teaching and research. Besides this, he is an active member of New Zealand Ministry of Health Infectious Diseases, Australian Virology Society, etc. Not only this, he is also acting as editor of journals of high repute like Viruses. Well, his list of achievements is going on and on. I will not go further, but one thing which I want to share with you about Matloob is when he's not dealing with the viruses, he enjoys swimming, jogging, and cooking. So Dr. Matloob will be deliberating on the topic viruses, pathogenic organisms of our ecosystem, and their interaction with the host. May I now request Dr. Matloob to deliver his talk. Over to you, sir.
Hello, everyone. Can you hear me all right? So thank you, Dr. Indu Mehta, and I really missed the paneer dosa from Chandigarh. So I love eating, and I can't find it here. I can't cook, I can't make it, but anyway. So I think it's a great forum, and it's really exciting. And I was just listening patiently to Dr. Misra, and I thought she gave an excellent talk, and she gave a lot of insight and all the way through from her early days in research and now as a kind of an entrepreneur. So first of all, I would like to thank the principal, Dr. Thapar, for introducing us, and also Indu, with whom I did my PhD in Chandigarh at MTech. And we were on the same floor. She was senior to me, but nevertheless, we got along really well, and it was a great fun at that time. Gosh, it was like a long time ago. I can't even remember. So I really would like to thank Indu for reaching out and inviting me and Dr. Misra to give this webinar and share our own experiences. So what I would like to do, my talk is actually, is in two parts. One part is, I thought it's a good idea to give an introduction to viruses, very basic introduction, because we have a mixed audience, right from starting from BSc first year and then potentially to master students. So I'll divide my talk in two parts, and one first half is basic virology and what are actually viruses and why do they matter the most. And I don't really have to emphasize that because we are going through a kind of the worst pandemic from a virus in the last 100 years. The last worst one was 1918. I was not born then, and I haven't seen that, but that's what they say. That was the worst one, and this is the second worst in the history of viruses. So let me just share my talk. First, the basic one. Okay, so yeah, so can everybody see it all right, Indu?
Yes, perfectly fine. It's
Okay,
visible.
good. Yeah, good. So as you can see that on the top left corner, there's so many cartoons of viruses. So basically, this cartoon shows that they're not a single virus in the ecosystem, and viruses are actually part of our ecosystem. A lot of people say we need to eradicate, eliminate, and all that. Well, you can actually eliminate one or two viruses, but a lot of them and plenty of them will remain part of our ecosystem. And the reason I'm going to explain to you in part of this talk. So just to go completely basic, just to ask questions, what are actually viruses? So when somebody asks you for a scientific definition, somebody who understands biology, so the definition of viruses are actually acellular microorganisms which require other organisms to survive. So basically they are parasites. Okay, they cannot survive without other organisms. And other definition could be, again, viruses are acellular microorganisms which require living cells to multiply. So they are obligate intercellular pathogens. And some people call them parasites. I call them pathogens because pathogens means viruses always almost end up causing a disease, and they're almost always fatal or harmful to the host compared to other parasites, because some other parasites, they just live within the host or alongside the host. They don't really harm them. It's kind of a symbiotic relationship. And to do that, to multiply, viruses utilize much of the machinery of that organisms they are infecting to multiply and eventually cause disease. So because of this characteristic, they are at the interface of living and non-living. So you must have read in your textbooks that viruses are non-living or living, but they live at the interface. So they're both. They're living because they can self-replicate, because they have nucleic acid as genetic material, and that nucleic acid could be RNA or DNA. They produce offspring, means they can reproduce, they can make progeny virus. So that's the characteristic of a living organism. And they also encode enzymes. So enzymes, as you know, are an important part of physiology to basically get the physiological functions going, and so that your metabolism can get going. So viruses, they can actually encode enzymes, particularly like polymerases, to replicate the nucleic acid. But at the same time, they also have certain characteristics of non-living organisms. For example, they don't have a cellular structure. It means like their structure is not defined like a mammalian cell, so like a eukaryotic cell or prokaryotic cell or archaeal cell. So they have a non-cellular structure. And they have obligatory dependence on another organisms for reproduction. Although they have a genetic material, but they cannot use on its own. They need other machinery from the other organisms to reproduce. And they do not possess their own metabolism. They do have enzymes. They can help metabolite the substrate, but they don't have their own machinery, for example, cytoplasm, nucleus, or plasma membrane, because of their non-cellular structures. So that's why they are non-living. So they live at the interface of living and non-living. So when you go all the way back in the history of diseases, and you look and you trace the history, viral diseases are actually ancient. The first description of a viral disease, which is foot and mouth disease of animals, was described all the way back to 1546 by an Italian monk. Oh, I can't really pronounce his name, so you tried. And the other one is polio. I mean, we all know about polio, and it's almost eradicated, but we still have pockets of polio, virus infecting people in parts of Pakistan, Nigeria, and even in India. And the first clinical description of this was found in 1789 by a British physician, Michael Underwood. And I hope you know about smallpox. So although this virus have been eradicated in late 1980s, but this is one of the worst viruses faced by the humankind. So its description was first described by Edward Jenner in 1796 through vaccination, what they used to call at the time variolation. So the vaccination, the strategy, and the concept, the fundamentals of the vaccine, derived from the smallpox discovery. And these viruses were actually, these diseases were actually known before even the causative agent virus was actually known or identified. And until they identified the mosaic disease of tobacco. The tobacco mosaic virus was the first virus to discovered around 1857 by some students, and then later it was characterized. And then the rabies. We all know rabies, generally spread by dog bites. And the causative agent of rabies was described by Louis Pasteur in around 1880. So these diseases were known, but it was not known they were caused by viruses until much later on. So this is a slide indicating the various shapes and sizes and various flavors of different viruses. And this is the culprit right now, the coronavirus. One of the coronaviruses that has been, causing the pandemic, and a number of cases keep rising and rising. And other things you can see that they come in different sizes and shapes. One of them is just pretty much like a spaceship. This one is like a bullet, and this is like a rod, and this again like a starship or something like that, like a crystal or something like that. And the smallest size could be 17 nanometer, and the largest one could be 1 micrometer. So they come in different shapes and sizes. So where are actually they found? And the simple answer is, where you find life, there you will find viruses. So wherever life exists, the virus will exist there. And it was estimated that viruses are the most abundant biological entity on Earth. And if you want to read more, I believe they are recording the slides. You can read this article about if you want to know more. And they're estimated about 10 to the power of 31 viruses on Earth. So I can't really even say that, like, whether it's a million or billion or trillion. It's like a 30 trillion or something viruses on Earth. And if you want to know more about, you can read this article. So meaning there may be 100 million times more viruses on Earth than there are stars in the universe. So now you can go and do your math, and you can imagine how many viruses are out there. Interestingly, I don't know, have you ever seen a movie? Hollywood movies are very good at predicting things and like aliens and all that. So lately people are asking question whether viruses are extraterrestrial, means they live in space. Is there a planet other than Earth where viruses exist? Well, soon we will find out, because there are so many companies they're planning to colonize or put people on other planet. Recently, NASA actually officially started exploring this option. I'm not sure whether aliens exist or not, but they're looking for them. So they officially started to look for viruses in space as well. So watch this space. You might find out soon. Now, so which organisms viruses infect? As I said, where you find life, you'll find viruses. So pretty much all cell types. If you go to your textbook, basically there are 3 domains of life exist: eukaryota, prokaryota, and archaea. So, and they have 3 different cell types, because these domains are defined based on the cell types. So viruses infect all these 3 cell types. So in other words, viruses infect all forms of life, not just humans, animals, plants, bacteria, fungi, and all that. And yes, interestingly, viruses also infect other viruses, and they are known as virophage. Very, very interesting organisms to study, and very interesting to find out about them, not just superficially what we know, what general public know about viruses. Now, just some terms I'm going to be using in my talk later on. So the organisms a virus infects is called a host organism. And basically there are 2 types of host. Clinical host means a host after infection, they can actually display symptoms. For example, if you get say right now, you get COVID-19 infected by SARS coronavirus 2. So the symptoms are you get dry cough, you get fever, you get chest pain, and some people also get nasal secretion. So these are the symptoms. And so basically, we are clinical host of many of these viruses. But there are other hosts out there which actually don't show any symptoms. They are called as reservoir host or intermediate host. For example, bats, birds, and mosquitoes. They just transmit the virus from the one clinical host to another clinical host. They don't show any symptoms. They don't show any disease, and the infection is not lethal. Okay. So now I need to hydrate a little bit. So that was about viruses, but how do they actually transmit between hosts? Obviously, there are so many viruses out there that different mechanisms they use transmit between the hosts. So the first one, which actually everyone can relate to, is the by respiratory droplets and secretions. This is called aerosol transmission, which is what is going on right now with SARS coronavirus 2. And the other one is the contaminated food and water. Remember, we all get diarrhea, we all get food poisoning, and said that food poisoning and diarrhea not is only because of some bacterial infection or fungal infection. It can also be through viruses, and 2 very common viruses which actually disseminate through this this process is rotavirus and norovirus. You must have heard about norovirus. Causes a lot of diarrheal outbreak on cruise ships. Third one is direct contact with bodily fluids like blood and semen, and classic example is HIV and hepatitis C. And also with direct contact with infected animals, for example, bats or birds, like and classic example for this avian influenza virus, which is, I think right now there are some outbreaks going on in India. And the lastly is vectors, like mosquitoes, aphids, flies, and nematodes. This is called vector transmission. And the classic example of this transmission is dengue, which is spread by mosquitoes. And India has a lot of problem with dengue, especially around Delhi and Mumbai, I believe. So that was between hosts. What about within the host? So basically, how viruses go inside the body of the host and disseminate within the host. So it depends on the mode of transmission. Obviously, if it's an aerosol transmission, you will inhale it through your mouth or your nose. You touch the object, contaminated object. For example, you shake hand or you touch the contaminated surface, and then you touch. We all have tendency to touch our face. So then the virus will, respiratory virus, that is, end up in your lungs and then start multiplying there. On the other hand, viruses which actually transmit through mosquitoes, for example, you get the mosquito bite and then it will go directly into your bloodstream. So this is called viremia. And then after the viruses will multiply inside the host body, and then they will release their progeny, and that progeny will come out in the blood, and then that called secondary viremia. And you must have heard about the systemic infection. Systemic infection is virus has actually, or for that matter, a pathogen has spread throughout the body. So that's called systemic infection. And some pathogens, including viruses, they can actually go into the brain and infect the brain. For example, the classic example of symptoms are like the meningitis and encephalitis, infection of brain by viruses like dengue. So these viruses called neurotropic virus, and the phenomenon is neurotropism. So that was about the transmission within the host. What about the disease? Like what kind of diseases viruses cause? Obviously, they're based upon mode of transmission. And again, number one is airborne diseases, for example, caused by influenza virus and SARS 2.0, SARS coronavirus 2. Influenza causes flu, and SARS causes COVID-19. So a lot of people get confused with the COVID-19. Said that, so the COVID-19 is the disease, coronavirus disease 2019, and the causative agent name is SARS coronavirus 2. In other words, severe acute respiratory syndrome coronavirus 2. Second one is food and waterborne diseases. For example, diarrhea caused by rotavirus, direct contact diseases like AIDS caused by HIV, and then arthropod-borne diseases, like mosquito-borne diseases, for example, dengue, and zoonotic disease like rabies. So rabies is spread through the bites of dogs. So the virus is actually is there in the saliva, replicates in the salivary glands of dogs, and when dog bite, for example, humans, and then they will transmit the virus to muscles, and that's how the virus will spread throughout the body and become neurotropic and cause the encephalitis. It's a neurotropic disease. So that was a bit of an introduction about viruses. I hope you get the idea that how fundamentally involved the viruses are in the ecosystem, and how they are, their interaction or their relationship with the host is critical, and that's why we end up getting a lot of diseases. Now, we all get common cold, for example, like sneezing and sore throat, viral diseases like flu, diarrhea and measles, chickenpox, all these virus diseases, but they are not as dangerous. You can get it, and then you can clear the infection, and after 5-6 days you're fine. And the vaccines are available against many of them, like hepatitis B, chickenpox, and measles, rubella, and all that. And pretty much everybody whoever was born after 1990, they must have been vaccinated against these viruses. So good. However, every often and then a new virus will emerge, and will create a havoc. Like right now, SARS coronavirus emerged and caused a pandemic. So how does it happen? How viruses emerge and end up causing such a deadly disease and such a large number of population getting infected? So when you go at the definition that about the emerging infectious diseases, basically the definition is of a new disease that has newly appeared in the population. Basically, that was new, nobody was infected before, and suddenly it appeared in a population anywhere in the world, or that has existed before, say 50 years, 60 years ago, and there were no incident of that particular disease or infection in the last 60 years. Then the classic example is Zika. Zika virus was not seen after 1951 or 2, and then suddenly appeared in 2015. So other one is they have existed before, but not seen for a long period of time, and then suddenly you start seeing their infection and rapidly increasing in the incidence. Okay. And this is the list of some of the major viruses which has emerged or re-emerged in humans during 20th and 21st century. And I already mentioned 1918 flu pandemic. So that was caused by influenza A virus H1N1 strain. It was emerged in USA and spread all over the world. At that time, World War I was going on, and there was a lot of movement of ships and people, not people, mostly army soldiers. They were traveling through ships and everything, and they took the virus from one place to the other. And then the second most significant virus emerged was HIV-1 in 1981, again in USA. Although its origin actually traced back to Africa, but the first cases were found in Los Angeles, in USA, I believe, Los Angeles, or maybe New York. And the third significant was, again, influenza H5N1 in 1997 in Hong Kong, and then SARS coronavirus 1. So not many people remember about this virus. So SARS coronavirus was emerged in Hong Kong in 2002, 2003. I was doing my postdoc at that time at NIH. So that was the very significant virus and caused about 800 people, 800 deaths and about 10,000 cases. So at that time, we had the opportunity to continue studying this virus, develop a vaccine 20 years ago or 18 years ago. So, but what happened, SARS coronavirus appeared and disappeared very quickly within 12 months. So people thought, oh, it's gone. Yeah, never mind. You know, we don't really need to do anything about it. And so they just sit back and relaxed. And 18 years, it was not inactive. The virus was still spreading among bats. So among bat population and other animals, it was circulating. But luckily, it didn't jump into humans. But then suddenly in 2019, the virus changed a little bit and then jumped into humans, and now we have the result. They call it SARS coronavirus 2 because the difference between SARS coronavirus 1, which appeared in 2002, and the one is spreading right now, it's very minimal. There is an 81 or 83% nucleotide similarity between the genome of SARS coronavirus 1 and 2. So now you can imagine that how close these viruses are. So in hindsight, looking back, if we would have developed all the vaccines and resources at that time, we would not have this kind of devastation which we are facing right now. Anyway, moving on. In 2009, another influenza virus emerged, caused the pandemic in 2009. Many of you will be aware of that. And swine flu, that they call it swine flu, it's still spreading in India. And the next coronavirus emerged in 2012 in Saudi Arabia. It was also very lethal and caused a lot of deaths. And then another influenza appeared in China, H7N9 in 2013. Ebola, you all must have read the news reappeared in West Africa in 2014 and caused about 15,000 deaths. And then Zika virus appeared in Brazil in 2015 and is spread by mosquitoes. And at that time, you must have heard or read the news. The classic feature of Zika virus infection is a disease which the babies, they had deformity in the head. Their brain was underdeveloped. It's called microcephaly. So viruses do cause neurological symptoms and deformities. So that excellently relate to what Kamna was talking about in her talk. And finally, SARS coronavirus 2.0. I call it 2.0, like that Rajnikant movie 2.0. So it emerged in China and is still ongoing. So what are the factors which drive the virus emergence? So we have 4 factors: ecological, human demographics and behavior, international travel and trade, and virus change and adaptation. So this is pretty much self-explanatory. If you put humans in contact with the natural host of a virus, then there will be a transmission and the virus will jump from animals to humans. And the classic example is the agriculture. For example, a lot of people do integrated farming. So you have a farm, you're growing, chickens there, you have pigs there, you have humans working on the farm. And both chickens and pigs are reservoir host, natural host of influenza virus. So the influenza virus can easily jump from poultry to pigs and pigs to humans and vice versa. So that's why in the previous slide I have shown you a list of viruses which emerge, and majority of them were influenza viruses. So influenza is still a very significant virus, and it still matters. People are not talking about influenza right now because obviously we have a bigger and more dangerous virus to tackle right now, SARS coronavirus 2. Another example is like if you make changes to the water ecosystem, you can breed mosquitoes, and that can carry the viruses like dengue and Zika. For example, stagnant water, not proper sanitation in metro and bigger cities. People live in small slums and all that, and so they will breed mosquitoes, and the mosquito will be infected by these viruses, and they will spread. Another important issue is climate change or global warming. I wrote an article last year about viruses and pretty much explain everything what I'm explaining to you. Potentially the next pandemic will be caused by viruses in conjunction with climate change. And the reason I'm saying it, because, for example, in New Zealand, the climate is temperate, so many mosquitoes cannot survive. But then if there's and like, for example, Norway or Denmark or Scandinavian countries, where its temperate climate is very cold, so many mosquitoes or many hosts of the viruses, they cannot survive. But what if there's global warming? What if there's climate change? Then these mosquitoes and other hosts, they will start migrating there and can survive. Birds, which are the reservoir host of flu, they can migrate from one continent to another. So in the next 50 to 100 years, this will be another challenge to look out for. And then lastly is flood. For example, if we have flood, so that can disseminate viruses like rotavirus and norovirus can cause diarrhea. And then the second factor is obviously human behavior, human demographics. That's why we had lockdown. Basically, if you lock down people, you reduce the number of hosts of the virus, and then you reduce the transmission. And that was the main reason of the lockdown. Lockdown won't necessarily save you from the disease or something like that, but it will limit the number of hosts virus can access. So basically, if there is a higher population density, less immunity, higher immunity, and so less immunity. Population density is high, so host is always available. So basically, you will have a stable virus-host interaction. So if one person is infected and another person is next to it, very in close proximity, the virus will infect that person. So you will never be able to eliminate the transmission cycle, and hence the lockdown. And obviously urbanization, if you have a mass movement of people carrying virus to new host locations, and then that virus will emerge in the new location. This one I don't really have to explain. We are all adults here. Human behavior is also important, which is spreading the virus. Intravenous drug use, if you use the same syringe to inject, multiple people. If one person is infected with HIV or hepatitis C, you will end up infecting, other people, and then multiple sex partners. Obviously, I don't know how many number of people are there in this photo. Just a joke. And this one. This actually happens a lot. So, getting too cozy. In this case, people are actually kissing pigs. Pigs are they are infected with influenza, and even camels are infected with MERS, Middle East Respiratory Syndrome, coronavirus. So these practices, people should refrain from doing that. So this could be the cause of the virus spread. And this is actually pretty obvious. We all know we live in a globalized world. People travel a lot, we go on vacation, to meet friends and family living, for example, in the US or New Zealand or Australia. So what happens that because of this globalization, we diminish dead-end virus-host interaction. Right? So dead-end virus-host interaction means, like, for example, if somebody got infected in Africa with a novel virus, and if they travel from Africa to US, now within 12 of, they have flights like within 17 hours, you could cross continent and end up in New York, all the way from, say, Hong Kong. So within 17 hours, an incubation period of a virus is less than 2 days. Some viruses cause symptoms very quickly, like within 24 hours, for example, flu. So means the dead end is not there. By the time you reach New York, you can infect other people. And look at the scenario 100 years ago. There were no planes, no faster transportation method. So at those times, people used to travel in ships. So somebody took the route of the ocean and travel from Africa, say, for example, to USA, and it used to take about 4 to 6 months to travel through the water. And somebody got sick in Africa, and they traveled through the ship, and they won't make it to the New York through the ship. They will die during their journey, and then they will end up in the ocean. So it means dead end. The virus host dies, virus dies. So if you want to get rid of the virus, you have to get rid of the host. That's a simple answer, but it's not possible, obviously, because we can't actually eliminate the human population just to eliminate the viruses. So diminishing dead-end virus-host interaction because of the globalization. That's why viruses transmit very, very fast. And this one, the last one, is the molecular change in the virus-host interactions. Once the virus infects a new host, it can adapt, and the adaptation can involve multiple changes within the virus as well as within the host. For example, there could be a genetic change in the virus structure, and genetic change in the genome or the protein, and that protein is better adapted to multiply that virus within the new host. And for the same reason, virus can adapt to an antiviral drug. They can change the target, and they can acquire the drug resistance. So this is a molecular change, and I will explain to you a little bit more in my second part of my talk. Although not all bad news, we do have sound prevention and treatment, methods to control viruses. We can actually prevent infection by surveillance. We can look out in which part of the world new infections are spreading, and this is the job of WHO. WHO has a very active surveillance program throughout the world, and then we have vaccines available. against many viruses like chickenpox, for example, and also treatment, antiviral drugs against influenza and HIV and hepatitis C. We have antibody therapy. You must have learned a lot during these times about antibody therapy, and also interferons is also which interferons are the molecules made by the host, for example, like us, to defeat the virus. We have a defense mechanism inside our body which can actually repel or block the infection. But obviously viruses are stronger than what we have, and that's why we end up getting the disease. Now, so virus-host interaction. So that was, I explained to you a little bit basic information about viruses, but how actually viruses enter the host and enter the host cell. Remember, every organism's, it has a cell, whether it's prokaryotic or prokaryotic, mostly single cell organism, mammals, and like us, we have multicellular, organization. So first virus enter the host, and then it will target the host cell. If it's a respiratory viruses, it will target the host cell in the respiratory tract. If it's a, say for example, diarrheal virus or causing diarrhea, it will target a host cell in the intestine system, in the digestive tract. Okay, so first, after entering the more complex host like us, the virus has to find a host cell, and then once it finds the host cell, it will attach. It will interact with the host cell, and then this will start the cascade of the infection of the host cell. Okay, so basically in the life cycle of a virus in a host cell, the 5 main steps. So the first, the virus particle will attach to the host cell. It's called attachment process or adsorption, and then the virus will enter the host cell. It will release its genetic material inside the host cell, so that genetic material can start the replication process. And then after the replication, it will synthesize the viral components. For example, copy of the genome and the proteins. And then once the enough raw material is made inside the host cell, then that raw material will come together and assemble a viral progeny, more virus particles. Okay, so that process is assembly. And then once the virus is assembled, it will release from the infected cell and then infect new cells, neighboring cells, or new host. Okay, so these are the 5 main steps in the virus in this life cycle. So this is a diagram of a eukaryotic host cell. If you can look at here, so the virus will enter, and the eukaryotic host cell is divided into and there's a plasma membrane, you have a cytoplasm, and you have a nucleus. So some viruses, they will enter the host cell cytoplasm, but only replicate in the cytoplasm. They will replicate, they will synthesize their component and assemble and get out. They won't go into the nucleus. So think of this life cycle as this is your house, and think of virus as a thief, a burglar entering your house. So viruses are thief. They will thief will come inside your house. It will go to your drawing room, living room, and then if it finds whatever it needs in the living room, it will take it and go back. So that's the analogy of the cytoplasmic replication. But if it needs what it is in your bedroom, for example, then it will go in the bedroom, for example, nucleus, and then replicate there, and then take it, and then get out. Okay, so it's the thief entering. Just like thief will come into your house, it might take your clothes, your money, your jewelry, and might eat your food and everything. So basically, empty everything and then leave you without everything. So this is the situation, analogous situation with the infection of a host cell. Okay. So we work on influenza virus. So I'll tell you a little bit about my research on influenza virus, what we are doing. So this is the structure of influenza virus. So this is the electron microscopic image. So this virus has a membrane and has a genome inside. The interesting aspect of this virus and unique aspect of this virus that you must have learned in your textbook, that the nucleic acid is linear or circular. Well, in this case, it's segmented. So you have individual 8 segments of this virus. So this is unique part, and this unique characteristic helps the virus to evolve and naturally into a new strain and all that. And this is the life cycle of influenza virus in the host cell. It will interact with the plasma membrane, enter by endocytosis, uncoat its genome, and that genome will go inside the nucleus and replicate, make mRNA and more genomic RNA copy, and then mRNA will come inside the cytoplasm, because remember the protein synthesis occurs in the cytoplasm, and then it will synthesize the viral proteins, and some of those viral proteins will go back inside the nucleus, replicate the genome, and then assemble what went in and assemble it, and what went in, and the same thing will go out and go to the plasma membrane. All the components will assemble on the plasma membrane, and the virus will assemble and release by neuraminidase action. And then when the virus particle is out there in the respiratory tract, it will infect other cells in the respiratory tract, the same host, and also breathe out. You know, you will sneeze and you will cough and you will throw these virus particles into the environment, and if somebody else inhales those droplets, and they will get infected. Okay, so that's the life cycle of our influenza virus in the host cell. So that was a bit of a background about viruses and how they behave. Now I'm going to talk about a little bit about the work we are doing in my lab. So I will switch, I will start to a new window. So I hope you can see this new window I'm starting. So as I explained to you that the viruses will, some viruses will replicate in the cytoplasm, and some viruses will replicate in the nucleus. So for example, influenza virus utilizes all 3 compartments or 3 parts of the cell: plasma membrane, cytoplasm, and nucleus. And a host cell has thousands and thousands of thousands of their own protein. So think of host cell is a big mall, right? Big mall has so many shops, so much going on, restaurant, this and that. And when you suddenly enter to the mall, and then you get bedazzled, okay? So it's a big environment. So think of one virus particle entering a host cell. It's a big environment for the virus particle, and it's very hostile, because virus is a thief, it's an invader. Host cell is not going to like it. So the host cell environment, for example, proteome, it's a very complex system, okay? So that complex system, the virus has to deal with that complex system, either to exploit or to antagonize. Means if there are some host component inside the cell, they are against the virus, virus has to overpower them to complete the life cycle, which I explained in the previous slide. So so this is the host protein is a very complex. I don't want to go into much detail, but one part of the proteome is important, which we focus on to look at the role of that part in influenza virus life cycle, is the protein modification. You must have read in your textbook that proteins are phosphorylated, glycosylated. So these are modifications. So protein polypeptide, it synthesizes a linear molecule, and then it will be modified by phosphorylation and glycosylation, create multiple species. Okay, think about like a clone. For example, Spider-Man. You know, one was the good clone, another one was the bad one. I don't know how many of you have seen that movie. So one of the protein modifications we focus on is acetylation. So it's the addition of acetyl group to the polypeptide, and for a particular function of that polypeptide. Interestingly, acetylation exists in all 3 cell types or 3 domains of life, eukaryota, prokaryota, and archaea. So we focus on the role of acetylation in influenza virus life cycle. So that's a bit technical, but I want to give you a snapshot of our research, what we are doing. I'll try to explain it in a simple way. So acetylation, obviously, it's a process of the protein modification, and any process, any changes in the protein is actually catalyzed by a set of enzymes. So histone acetyltransferases is a misnomer because it was first identified in histone protein. So acetyltransferases, which add the acetyl group to protein and acetylate. But there are a group of enzymes which actually remove the acetyl group. They call it deacetylases. So remember, pretty much all the enzymatic reactions are irreversible. So, and on both end, reversible, both end the enzymes would be involved. So acetyltransferases will add the acetyl group, and deacetylases remove the acetyl group. And there are about 18 of them known, 18 acetyltransferases known, and about 18 deacetylases known. So acetylation plays actually a very important role in the homeostasis, in the normal physiology of the host. And if there is a slight imbalance in the physiology, in the acetylation, then you end up getting a disease like cancer or neurodegeneration or deformity or something like that. So it's a normal phenomenon. So you need a balance in this. And when the viruses infect a host cell, a normal host cell, there is no such thing in normal situation. Viruses, for example, a thief will enter into your house, and if you happen to be in the house, it will be a chaos. You will be yelling, you'll be, shouting, thief is trying to scare you or something. So that kind of situation. So there is a lot of imbalance going on when the infection occurs. So what we found a long time ago when I was in the United States, if you treat the cells, infected cells, so we use a cell culture system, we can grow the cells from human lung and then infect, in the lab. I'm sure you do practicals and you know about it, what I'm talking about. So when you infect normal cells and then you treat the cells with some sort of inhibitor or some chemicals which will imbalance the acetylation process. So when we infected the cells and treated them and to induce the acetylation environment, there was a influenza virus infection increase, means number of progeny released increase, means acetylation actually is helpful, is helping the virus to replicate. So that was a very important finding around 2011, 2010 when I was in the United States. So that at that time, it was the first time somebody has discovered something like this in influenza. So now going back to the acetylation. So the acetyltransferases and deacetylases are central regulator of acetylation. So if the acetylation is important for virus infection, then the enzymes which are adding the acetyl group to the proteins in the host cell, they will be proviral because they are helping to induce the acetylation environment. On the other hand, deacetylases, they will remove the acetyl group. They are working against the viruses. Okay. For example, if you're eating, say, paneer dosa and you like it, and somebody snatch it away from you, you don't like that. That is anti-you. Or if you're eating paneer dosa and somebody give you more to eat, that's pro-you. That means it's helping you. So that kind of situation in this, acetyltransferases are pro-viral, pro-influenza virus, and deacetylases are anti-influenza virus. So this is the theme we developed in our lab. And remember, there are at least 18 of both families are known. And so we started working on what is the role of individual histone deacetylases, in short, HDACs, in influenza virus infection. So obviously, hypothesis was they will exhibit anti-influenza viral properties. And we actually hypothesized in our lab, and also one lab in Princeton, New Jersey. They also work on this particular family or class of HDACs, and they show that they have anti-influenza virus properties, and these are the references for the publication we published. So I'll just give you one example of what I'm talking about. So we will focus on is class 2 HTECs called HTEC4. That what is, how do we actually define that this particular HTEC has anti-influenza viral property. So in this case, we grow the cells, and we can actually genetically manipulate the expression in the host cell. A lot of it was common. I was talking about there's so much advancement in the technology and everything. You can do a lot of things. You can use multiple techniques to actually address or answer one problem. So in this case, you don't really have to go understand the whole thing. This is a technique we use called Western blotting to see the status of manipulations we are making. So in this case, we are actually depleting using a technique called RNA interference. We are actually depleting the expression of HTEC4, and then after depletion, we are infecting those cells with influenza virus, comparing it to the control to see what's happening when we deplete the expression of HTEC4. So this is the confirmation of the depletion, that we can successfully deplete it, and this is the confirmation that when we deplete it, the virus infection goes up, and the data is statistically significant. On the other hand, basically, if you had $10 in your pocket, and somebody take those away, that means they're depleting you of money, so you can't do the function. So this is basically similar to that. What about opposite occurs? If somebody gives you another 10 rupees or $10, then you will have 20. So whatever function, whatever things you're going to purchase with those 10 rupees, then you have 20 rupees, so you can purchase more. So what we did here, we manipulated the cell such a way that it expresses the quantity of HTEC4 even more what exists in the normal scenario. So then the virus application went down. So that this explains that HTEC4 has anti-influenza property. Then what is the mechanism that, although it has anti-influenza property, that what is the mechanism that HTEC4, through which method is actually exerting anti-influenza property? For example, if you want to go to Delhi from Chandigarh, so there are multiple mechanisms, multiple ways to go to Delhi from Chandigarh. You take the Shatabdi Express, or you take the plane, or you take the bus. So we wanted to know through which mechanisms HTEC4 is actually exerting its anti-influenza function. So I explained you, I told you a little bit about that we have, our body has innate defense. So whenever a pathogen like virus or any invader enters the host, the multiple pathways get activated. And a classic example of this molecule called STAT1, you can see that. So this is a time course of the infection in the host cell. And if you can see, when we infect in the normal scenario, this after like 6 hours of infection, you'll see the expression of this molecule, which is ready to defend the cells or host against the virus. So, and there are other molecules as well. You don't have to go into that, but you can read this. It's already published. It's on my website. So we found out that HTEC4 is part of this innate immune response. So if you can focus in here on this one here, and it's more clear here, when HTEC4 was not there, the innate immune response of the host was compromised. So that's how it was, playing the anti-influenza virus role. And this is further data, quantification, and all that's confirming that. Now, obviously, if a thief enters your house and you fight back, so thief is more strong because they have the intention to enter your house, so they will have some sort of weapon with them, so they are well prepared. So in this case also, influenza is not going to sit idly and let HTEC4 antagonize it. So what influenza does, it actually fight back against HTEC4. So in this case, we are showing that HTEC4 level are actually down-regulated in the infected cells because it's anti-influenza, because influenza does not want the normal expression of HTEC4 being expressed after the infection. So these are uninfected cells, and these are infected cells. So in other words, influenza also antagonizing HTEC4 so that it can successfully infect the host, and that's why we end up getting the flu. But this is the mechanism we are trying to understand, that how many pathways influenza is actually, engaging with the host. So this explains that influenza is actually successfully antagonizing HTEC4, and this is multiple pathways, it utilizes, and basically this apoptotic pathway it's employing to antagonize HTEC4. So what weapon of its own influenza using? For example, a thief will use a knife, a stick, or gun or something like that. So we wanted to know what weapon influenza virus is employing to antagonize, to overpower HTEC4. And so again, in this case, we individually depleted the expression of individual influenza virus gene. And so when we depleted this particular gene, and when it was not there, there is a recovery of HTEC4 here. So that gene is called polymerase acidic, which is part of the RNA polymerase of influenza virus. So we confirmed that but it's highly technical, but I just want to give you a flavor of what kind of techniques and what kind of methods, like Kamna was trying to explain, that we use, and how far the science has come and what we can do. So we identified the PA gene of influenza is the weapon influenza using to antagonize HTEC4, which is an anti-influenza factor. But then we went further, and I don't know whether you know that the viruses are very clever organisms. They have only like limited number of genes. I don't know whether you know how many human genes exist. I mean, I hope you know that human genome has been sequenced, and we have about 21,000 genes. But viruses, they have only like 10 genes, 5 genes, 8 genes, and these 8 genes, they do all the work. And the way they do it, the viruses are very smart. They can actually express more than one protein from one gene. So this is the case in the case of PA gene. PA gene expresses more than one, and here we identified that the PA gene is expressing a protein called PAX, which is involved in antagonizing. So we went a bit deeper to identify the weapon influenza using to antagonize HTEC4. Right. So the next one. Finally, I'm in my last slide. So in this case, we identified HTEC4 as an anti-influenza factor, and by doing all this, exercise and everything, experiments, we wanted to further prove that host acetylation machinery plays an important supportive role during influenza virus infection, and maybe in the future, we could actually target this to treat influenza virus infections. But we are not there yet. We have to identify the role of other HTECs as well. So finally, I just want to acknowledge people who did the work. Henry actually did the work. He did this was his PhD project. He published the data. Now he has graduated, and these are the funding agencies which actually supported the research. So thank you very much. I'm sorry if I went over time, and I will be happy to take any questions.
Well, thank you very much. Now questions from the audience. Anybody wants to ask anything? Most welcome.
Good morning, sir.
This is Vanshika.
Good morning and good afternoon.
Good morning. Yes.
Hello, Vanshika.
Yes, sir. So my question was, I have just a curiosity to ask. Like you told about the two coronavirus, SARS Corona 1 and the one and 2.0.
Mm-hmm.
So what was the difference in them, that the first one, it lasted only for 12 hours, and the next one, the second one, 2.0, that is continuing till now?
What was the difference? I'm sorry, I did not understand the second part.
Sir, as you told that SARS Corona 1, the first, the one which appeared, that only lasted for 12 hours. That didn't last for a long time.
Oh yeah, so it lasted only for a short period of time because I think SARS coronavirus 1 was not transmissible efficiently between human to human, person to person. So the person to person transmission was limited. So that virus was kind of, the transmission of that virus was kind of at the interface of direct contact. So if you're in a very close proximity of the person who was infected with the SARS coronavirus 1, then only you will get infected. And that's why during that time, many people in the hospital, like nurses and doctors, they got infected with SARS coronavirus 1. And they quarantined very efficiently at that time. They used the same measure they're using right now. And that's how they were able to control the virus. And that virus was not like, the transmission is very critical for viruses to cause pandemics. So if you can control it, but this virus, again, emerge more efficiently. Means like, you know, like a villain, for example, in a movie, if you, in the beginning, you can overpower and then it will go to jail. So the villain will come back with more brain power and become more smarter. And then, so that kind of situation that the virus was not as transmissible. But then once it kept circulating in bats and other animals, and then it acquired that capability to transmit very efficiently between humans to humans, and then jump very quickly, and then it was very difficult to stop. That was one of the main reasons.
So any other question from the audience?
Yes, sir. Hello, Dr. Matlub.
Hello.
Yes, sir. My question is, as you are saying, there are certain vector hosts that are involved in the transmission of the viruses. So why not those vector hosts are susceptible to that infection?
Wonderful, wonderful, very, very good question. So that has some, if you understood the later part of my talk, innate immune response. So
yeah. Okay.
the innate immune response of those hosts, for example, bats, it's different to the clinical host. So viruses remain dormant in those hosts and do not replicate as efficiently. So viruses, for example, they also cause acute infection and chronic infection, latent infection. So they remain latent in the intermediate host because of the innate immune response is different. So the innate immune response, for example, bats or mosquitoes, is different to the clinical host. And that's why they remain there, latently. And as soon as, because they're not meant to, cause the infection, in those hosts, they just use them as vectors. So viruses are very smart organisms. They don't, express their gene in such a way they are recognized by the host innate immune response in those hosts. But as soon as those viruses jump from those hosts into clinical host, then they start, replicating and then start multiplying efficiently.
Okay, thank you so much, sir.
Okay. You're welcome.
Yes. Any other question?
Good morning, sir.
Good morning.
As we all know, COVID-19 has declared pandemic by World Health Organization, and so many cases were reported globally. So people are becoming more conscious about their immunity. Because obviously there is direct relation between the COVID-19 transmission and immunity. So with this concern of people, I think the scope of the immunology is somewhere increases, like for the purpose of studies. Am I right?
Yes, definitely.
For the higher studies, what is the scope of immunology, with your opinion?
Yeah, I mean, in my opinion, so short answer to your question is yes, definitely. There is a very high scope of immunology, innate immune response, designing vaccine and all that, antiviral drugs, and it's a great opportunity to pursue it. But I'll just go back a little bit. What I tell my students, I mean, there are two ways you can choose to study. One, follow your passion. What actually, that bulb, Chacha Chaudhary bulb in your brain, when you see something, when you say, for example, you're in Chandigarh, you go to Sector 17 or what is that other place you go to, Himachal side. So if you like, greenery or something, so that relaxes you, that excites you. So if you really like to study immune response against SARS 2.0, then you should definitely go ahead. I believe you're asking for your future study. You should definitely go ahead and study. But
Yeah.
if something else excites you, for example, SARS biology itself, fundamental biology, that how it replicates, the genome and how it assembles in the whole cell, then you go for that. But every field, in my opinion, as far as it's related to viruses, remember, viruses are part of our ecosystem. They're not going anywhere. People talking about some you can eradicate. So there are many, many opportunities, and there are many, many things we don't know about them, how they interact with the host, as far as, for example, immune response, innate immune response, their vaccination, how do we design vaccine, whether they're effective or not, antiviral drugs, and et cetera, et cetera. So yes, immunology, it's a very exciting field, but choose what excites you, what makes you happy, and what actually keeps you awake at night. Because sometimes I go to bed and I start thinking, and end up writing something. So I hope that answers your question.
Yes, sir. Thank you.
Anybody else?
Anybody else? It's a wonderful platform. You make the best use of this opportunity, I think, students.
Go ahead, don't be shy.
Good morning, sir.
Good morning.
Sir, I wanted to ask, in your opinion, is there actually any difference in doing PhD from India and PhD from foreign, like US or Canada? Research studies.
I actually don't think so, because India, when I left India exactly 21 years ago, I think India is different now, and even at that time, like India produced. I'm the product of it. I did PhD in India. Dr. Mehta did PhD in India, and I believe Kamna, Dr. Mishra also did PhD in India. So I think the standard of PhD in India is excellent. In my opinion, there are many, many. At that time, there were very good institute, but now I think number has increased manifold. I believe there are ICER in Mohali, and there are a couple of institutes in Haryana. That there's so many institutes are coming up in India. So there are many places in India, and of course, IMTech. We had a great time there. When I did my PhD. So again, I will probably piggyback on my question answer. I answer to previous question is look for the opportunity. Whether if you get the opportunity in India, and if it's a good opportunity, there are many factors you have to look at. Like, for example, if you want to stay in Chandigarh, then look for opportunities in Chandigarh which aligns with your interest. If you want to work on viruses or neurosciences or immunology and all that. But if you really want to explore the world, then you go out, you go overseas, Germany, USA. Well, right now USA situation is bit. Canada, New Zealand, beautiful country, COVID free right now. So no, I won't probably say that if you do PhD in India, it's not good, or if you do PhD from overseas, it's good. There are always pros and cons. But you just have to, because when we had the opportunity to look for a PhD or postdoc, at that time, remember, internet was just up and coming. We didn't have, websites and everything, social media. Basically, there was nothing. So we had very limited avenues to explore. But now, in current generation has, basically the whole world is in your hand, in your smartphone. You can search anything. So I would encourage you to look for opportunities which align with your interest and your future plans. So then, discuss with, for example, Indu, and with your peers and with your, teachers and lecturers, and then decide, and your parents, of course, and if you're have your second half then with them as well. Does that answer your question?
Yes, sir. Thank you so much.
You're welcome.
So, any other question from the audience? All right. Thank you very much. Hanji, Angita. Yes, Angita, it's your turn now. Please, bolo beta.
Sir, I want to ask, like you said that viruses, we can't eradicate them from our environment. And we have seen that it is quite harmful, like in the case of COVID-19. Is there any way that we can manipulate a virus so that it can be helpful to us? Like instead of a foe, a virus become a friend?
Excellent question. Excellent question. Yes, yes, we have been manipulating viruses. I don't know whether you have heard of oncolytic therapy. So viruses like pox viruses, which actually did my postdoc on, has been manipulated to kill cancer cells. So they are used in cancer therapy. And it relates to Kamna's talk. The virus is also being used in the therapy of neurological diseases like Alzheimer and recombinant viruses. You must have heard about recombinant viruses. Recombinant viruses are used to express proteins to make vaccines. So the viruses are actually exploited as for useful purposes as well. And biotech industry has taken a lot of interest in it. And so I can give you a lot of examples. Reoviruses, for example, they have been tried as oncolytic agents. Herpes virus have been approved in Japan and China, I believe, for a cancer therapy. Yeah, so I mean, the world is really, really open to the advancement. Unfortunately, you know, biological sciences or biomedical research or the achievement or the advancement or progress in this field is not advertised in the open platform like, say, Facebook or, you know, those kind of things, mostly technology. So there's so much going on in the world, in the world of science, and viruses are being exploited to make vaccines, to make, you know, express proteins for various purposes. Viruses are being exploited to express growth factors, for example, to treat skin diseases. And so, yeah, definitely, they are being exploited. And there is a huge field, you know, out there and opportunities out there everywhere, including in India. Does that answer your question?
Yes, sir.
Good.
So thank you very much for taking so many questions and answering them promptly. Thank you very much for giving an excellent talk. The talk was very relevant as the students study all these in their curriculum also. It was very informative and useful. We've been fortunate enough to listen to your valuable talk. Thanks once more.
You're welcome. I really enjoyed it. It was good, actually. Great forum, you know, to.
Yeah, absolutely.
Yeah.
That's absolutely.
I hope it was useful to other students as well who did not ask questions.
Yeah, definitely. I hope so. So, it's time for the vote of thanks now. It is my pleasant duty to propose a vote of thanks. It goes without saying that both the talks delivered by the eminent speakers were extremely informative, valuable, interesting, and rewarding. The webinar has been interactive and achieved its purpose. I'm sure that deliberations would be well utilized by the audience in the times to come. It is a historical event and would be remembered for all times to come. Credit goes to all the stakeholders. First of all, I express my gratitude and thanks to our worthy principal, Dr. Balraj Thapar, as well as Dean Sciences, Dr. Sajeev Soni, for encouraging me to organize this international webinar. I'm extremely grateful to them. Special thanks to Principal Sir for delivering his inaugural address. Thank you very much, sir. I'm grateful to Dr. Kamna Misra for having accepted my request and delivering such a crisp talk. which was deeply appreciated by the audience. It was very informative and useful. Thank you very much indeed. I'm also grateful to Dr. Matloub Hussain for his very lucrative and elaborated talk, which was of immense importance for the audience of this webinar. I take this opportunity to thank him from the core of my heart. The learned faculty members had been an important component of the audience of this webinar. Their presence today made the webinar all the more significant. I'm extremely thankful to all of them. I'm also thankful to the teaching and non-teaching staff of the zoology department for their cooperation and help. Last but not the least, I express my sincere thanks to all my students who made full use of the opportunity. Their presence was very important and apparently beneficial to them. I'm glad that they could make it. Thank you very much, my dear students. The webinar comes to the happy end. Thank you very much, one and all. God bless you all. Thanks a lot.
All right. Thank you. I'll go home now.
Yeah.
Okay.